target validation is probably the most important decision that we make, actually, in our industry and in R&D. The targets that you work on, the pathways that you work on, they start a trigger of events that's, can last 15 to 20 years from the very time you sort of start in the research phase all the way to hopefully getting through development and ultimately launching. So it's one of the most critical decisions we make, and I would say it's also a critical decision in academia, where you choose what areas to work in and how you validate your biology or invalidate the biology and the pathways that you're most excited about. What I want to do over the next 45 minutes or so is give a little bit of a flavor of the company and some of the shifts that we've made in our culture and the strategies that we've taken in Astra Zeneca, and then walk you through some case studies that will hopefully exemplify those approaches and that strategy, and then talk at the end about some of the technologies that I think are transforming how we do R&D, both in the pharmaceutical and biotech industry, but also, I think, in academia as well. And hopefully we'll have a bit of time for Q&A. So our strategy and how we've gone about reshaping our pipeline, and we really have been working very hard for the past, I joined the company in 2010, to reshape our strategy and our pipeline. I would say when I first joined the company and you asked external people about AstraZeneca as a company, if you're an investor, you basically would say, don't invest a single penny in AstraZeneca R&D because you're destroying value. That was what we kind of came into for those of us that joined the company 2010 and shortly thereafter. And today, I think the external view is very, very different. And that's been because we've spent a lot of time focusing on the science. Now, one of the things that we've done as a consequence of that focus on science is decide which areas we think we have strengths in and which areas we think we can't really compete. 2010, we were spread all over the place. And when Pascal joined, actually, our CEO in 2012, and he looked at all the things that we were doing, looked at the things that I'd been doing in the IMED, said, we've got to focus on 3 key areas where we have the critical mass, the scientific depth and expertise, the clinical experience, the commercial horsepower to absolutely drive innovation, novel therapies, and make a difference to patients. And those 3 areas were oncology, cardiovascular disease, metabolic diabetes, and chronic kidney disease, and respiratory inflammation, autoimmunity. Now, that doesn't mean that other areas are not important, that other people aren't going to work in different areas, but for us, as an organization, that focus became very, very important. And then investing in that focus to make sure that whatever we did at a scale and depth that would enable us to compete globally. Now, there are 2 other areas that we work in, anti-infectives and neuroscience, but we work in that in a quite different way. Smaller levels of investment, I would say, less than 10% of our total investment in R&D, of the $6 billion that we spend every year, is invested in neuroscience and infection. The large majority is in those 3 core areas. And then on top of that, the capabilities that we have, again, our traditional small molecule capability, the capabilities we've built through acquiring Cambridge Antibody Technologies and MedImmune in antibody engineering and biologics, and then more recently in devices with acquisitions of Almirall and Pearl, which enabled us to get pretty much any molecule into an inhaled formulation, which is fantastic for respiratory illnesses. That's enabled us to really build, I think, a depth and a capability that we feel very good about. Now, in terms of how we're organized, this is one of the easiest companies I've had the pleasure in working with, and as you heard, I've kind of experienced lots of different cultures and companies. We're organized in 2 biotech units. So I run one of the biotech units, Innovative Medicines Early Development, and Bahija Jalal runs the other one, which is called MedImmune. We're accountable for everything from target identification, so which pathways we work in, all the way to the end of phase 2 development. So basically, I have to hand over a phase 3 ready asset, which is late-stage clinical trials when you're getting to thousands of patients and really getting ready for a registration trial where you hope to get approved. So Bahija and I both have accountability for delivering these phase 3 ready assets. And the nice thing is that I don't decide whether something is phase 3 ready. Our head of global medicines development or late-stage development actually is the customer from me. And if he doesn't like what I'm providing him, it doesn't go forward. So that relationship becomes very, very important. And actually, who has the decision rights for when you move a molecule forward is important, because otherwise you tend to kind of believe your own press clippings, and you can bias the quality of what moves forwards. So it's very important that someone else is making the decision about what moves into late-stage development, not myself, who's handing the asset over, or Bahija. Ultimately, then, how it gets through late stage and commercialized. We're a very tight-knit group, one of the most collaborative groups of scientific leaders I've ever worked in, and we all sit on the CEO's executive team. And so the conversations we have at that senior executive team are very science-focused. And the great thing about having a CEO who has a scientific background is that we have much more focus on science and biology than I've ever had in any other company at that senior level. So we talk about projects, we talk about strategy, we talk about biology and pathways and targets, and we get as difficult questions from our CEO as we do from some of our scientists, which was actually very good. Now, one of the things that I did when I first joined the company was review all of the decisions that we'd made from 2005 to 2010 in terms of the types of projects that we ran, the way that we ran them, the decisions that we made, and tried to learn what we could from our history to enable us to look forwards. Because as I mentioned to you earlier, from a productivity perspective and delivery perspective, Astra Zeneca had not been a very strong company. It had historically before that, but 2005, 2010, not very good. Depending on how you measure us. If you measured us a number of widgets of things that we had done, we were fantastic. We were top 2. But if you measured us at output, number of successful phase 2s, phase 3s, launches, approvals, we were actually bottom 2. Now, what was and this actually was published in last year in Nature Reviews Drug Discovery. What we learned, and the bottom, if there's one take-home message that you take from this, it's that you have to focus your scientists on quality and not quantity, on what you do and not the volume of what you do. And the other piece is that you want your scientists to be really truth-seeking, to be trying to disprove their hypothesis. If you reward them for getting to the next milestone every time, they find ways for getting to the next milestone. So if you're recognizing candidates, a candidate that's ready to move into clinical development, and you reward them on the number of candidates they have to deliver, scientists are fantastic. They will deliver whatever number of candidates you want. If you reward them for the volume, for the quality of what they do, how strongly we believe or understand the biological rationale, how confident we are in the preclinical data we've generated, in the genetic validation of the target, that becomes a very different conversation when you review a program. So this is all about quality. We actually distilled it down to something that we call the 5 R's, and they're listed here. So the first one is actually about focusing on the biology. How well do you understand the target, the preclinical data, the preclinical models, the genetic validation, the human genetic validation of the target or pathway that you want to work in, and how well do you, and how rigorously do you question that hypothesis and challenge it to try and improve your understanding and not stop until you've ultimately got your drug approved. The second one is right tissue. And by that I mean, when you have a molecule that modifies a particular target that you believe in, is there a way of demonstrating that you actually engaging that target? So for example, if it's a receptor in the heart, how long do you need to engage or block that receptor for? How do you demonstrate that you've blocked it preclinically and then ultimately clinically? What are the biomarkers that you use? Do you understand whether you have to block it 80%, 70%, 50%, for 6 hours, for 12 hours, for 24 hours? We have to understand what that relationship is, how it translates to the human patient, and then ensure that when we go into the clinic and we ask the experiment, did we block the target for the right amount of time in the right organ, that we have a way of demonstrating that. You'll be amazed how many programs we ran in the clinic from 2005 to 2010, where we spent $50, $60 million in a phase 2 experiment in patients, and then you ask the question, did you test your scientific hypothesis? And the scientists and clinicians did not know, because they had no way of validating whether they engaged the target in question in the right organ for the right amount of time. We don't do that anymore. We don't move things into the clinic unless we can demonstrate target engagement early in development so that we know that something is worth investing in later stage development. Right safety. When you're incentivizing your scientists by volume, numbers of candidates, numbers of phase 1s, numbers of INDs, they're amazingly good at finding ways around safety signals. They lower the dose. They say, oh, the risk-benefits are fine, don't worry about it. My experience, when you have safety signals, they get worse through development, not better. In the same way that your efficacy signals get more noisy as you go into larger patient populations. So when you have a safety signal right at the outset of your program, that's going to get worse. So when you incentivize your teams to bring molecules that are of the right quality, where we're not going to fail in phase 1 and phase 2, the questions they ask early on in terms of the safety of the molecule are very, very different. They don't just try and lower the dose or avoid the questions, I'll worry about it in phase 1 or phase 2. They actually stop the molecule early and they move on to another molecule or another program. Right patients, one of the most important bits, and many of the case studies I'll show you highlight the importance of this. Defining the patient population that will best respond to your drug, whether it's an antibody or a small molecule, is absolutely critically important. The number of companies I've been at, and I would say this has changed hugely across all the companies recently in our industry, but the number of conversations I've had with commercial colleagues about, well, you can't work on that because the population's too small, where they'd rather have a drug that is a $5 billion peak year sales drug in all the population, even though it's never going to work. They prefer that, used to prefer that to having a drug that's going to make $200 million, but where you know that the science is really strong and genetically validated. That has actually gone out the window now. The question we ask is, which is the patient population that's most likely to respond? That's the one we test in the clinic. And actually, as long as we understand that, then we make sure that the investment we make is cognizant of the fact that it's a smaller patient population as opposed to a very large patient population. But we won't move something forwards again if we don't understand and define the most responsive patient population. It's going to give you the best benefit, risk, reward when you're doing your clinical studies. And that's important because when I talk about commercial, it's not about that it's a big peak year sales. It's about that I understand what the level of investment is for the size of the opportunity. So if it's a very small opportunity in a small population, I don't want to be spending $600 million in phase 2 and phase 3 to develop the molecule. I want to have a sensible investment plan, development plan that gets me an answer quickly and hopefully gets me to a regulator quickly. And again, I'll show you some examples of that. All of this put together changes the culture of a company because you're focused on quality and asking critical questions that actually not just we, but others have shown make a real difference to your success rates and to your ability to move a molecule from candidate selection through development and ultimately to approval, which is what we're all trying to do. Now, going hand in hand with this is the importance of doing great science, not just good science, but great science. One of the big misnomers that I get very upset about is when people say that in industry we don't do good science. Because we do great science, actually, and much of what we do, I think, can be compared with some of the best academic groups around the world. And when you look at the types of people that we're hiring and the publication rate of, this is just the IMED, this isn't all of Astra Zeneca, this is my part of the organization. This year we'll have published around 25 high-impact papers in Nature, Cell, Science, Cancer Cell, Genentech, that I think people recognize as being one of the most successful scientific organizations that are very science-led and has been doing it for the last couple of decades, for me is the exemplar of what good scientific leadership is about. We've been doing this for 2 or 3 years. You see where we were in 2010, we'd published 1 high-impact paper. The trick for me is now that we can do this sustainably year on year. This is ingrained into our culture, and publishing our science, sharing our data, which also makes you a much better collaborator, a much more attractive recruiter for great scientists, as well as for biotech partnerships, is an incredibly important part of what we do. So this misnomer that private companies, that pharma and biotech companies don't do great science, they don't publish their data, is wrong. Some do it more than others, but at least in Astra Zeneca, this is every scientist in our organization has to publish. It's part of our goals and part of our objectives. And all of this is translated from where we were in 2010, where we actually didn't really have a pipeline, to now where we have a pipeline that I think is pretty balanced across large and small molecules, across those core disease areas. You're not meant to read any of these things, but it just gives you a flavor. You can do if you want. You can tweet it as well if you want. It's a good pipeline. But you can go onto our website and see it in more detail. But we have a good balance between oncology, respiratory, cardiovascular, early, late-stage development. And the key here is that the flow is continuous. This isn't ever a static picture. It continues to flow. And the key measure of success is successful drugs approved where there's a high unmet medical need. That's how we measure our success, not by the number of candidates that we put in or the number of INDs that we file. Okay, let me take you through a few case studies to highlight some of the examples. The first one, and actually just to put this slide, because I think this is actually important and obviously relevant to the conference. The world over the past 5 years has changed quite dramatically. I remember when I started my career actually at Janssen, before I went to SmithKline Beecham, which became GSK, everyone was spending huge amounts of money, David will remember this, cloning ESTs from Insight databases and HDS databases. We all went EST crazy, including my group. And every EST that we sequenced and then tried to find the gene, sometimes we found a gene, sometimes we didn't, we patented it. And it was actually one of the biggest wastes of money actually out there. I mean, it generated very, very little value, and actually companies spent fortunes on these big database partnerships. Now, what has happened, though, I think, over the past decade, is with the advent of next-generation sequencing, our ability to actually do much deeper population-type genetics and rare variant genetics, it's really helped us validate targets in a very, very different way to how we were able to do in the 1990s. And that is going to continue to change and evolve, and I'm sure one of the things that you talk about over the next couple of days is how you handle all of that data, all of that information, both from the public and private domain, how you create bioinformatics groups and scientists that can actually engage that data and ask sophisticated questions to help validate or invalidate scientific hypotheses. John Dinesh, I think, is talking, will give a fantastic talk on little LP LA2. Well, let me start with one case. This is a drug called osimertinib. It was actually just approved about 10 days ago. The brand name is going to be called Tagrisso. It's moved so fast that we actually haven't had a chance to have a generic name, which is the osimertinib. It's always been called AZD1991. Now, this is an example of the power of really good target validation. This drug went from the first time in man to approval in 2.5 years, which is the fastest ever development of a drug in our industry. 2.5 years, unprecedented. And the reason it moved so fast is because it was based on absolutely phenomenal science and understanding the right patient population to go after in the clinical development plan. And I'll talk to you through this story. So if I take you back a little bit and give you a little bit of lung cancer history. So pre-1980s, a patient would come in with lung cancer, they'd be diagnosed by a pathologist, and you'd have different types of pathology of cell types in the tumor, and you'd be diagnosed as either being squamous, large cell, or having an adenocarcinoma. Move forwards to the 1980s, late 1980s, the first two oncogenic drivers of lung cancer were identified. Actually, the first one was identified, which was KRAS, a target that we still can't go after from a drug discovery perspective, although we're trying very, very hard. And it's still an incredibly exciting target if we could find a way of modulating it. And then not until early 2000 did the second oncogenic driver get identified, which was the EGFR receptor and various mutations and exon deletions that basically activated the receptor. And those activating mutations generated the first generation of EGFR inhibitors, Tarceva, gefitinib, which actually became mainstay therapy for lung cancer patients who had EGFR mutations. Now go forward just a decade to today and beyond, and you can see how lung cancer has changed in terms of defining subsets of patients with very specific genetic drivers of disease. Each one of those a potential novel target. So I mentioned gefitinib, or Iressa. This is actually the catalytic domain, the kinase domain of the EGFR receptor, and you can see how gefitinib fits into the pocket. It's a very effective drug, as is Tarceva, but as with most TKI therapies, target kinase inhibitor therapies, the tumors are very smart and are able to acquire resistance over a period of 6 to 12 months. And in this particular case, the resistance mechanism, which you see in about 70% of lung cancer patients, is a mutation which is called a T790M mutation, a threonine mutating to a methionine. And on the next slide, you can just see that little shift, right? One amino acid. That one amino acid change basically tosses out the gefitinib, prevents it from binding to the catalytic site, and makes the drug inactive, so the tumor progresses. Now, understanding that biology means that your chemistry team can find a molecule that is able to attack that new mutation. And that was what we did. AZD9291, or Tagrisso, or osimertinib, was discovered about 3 years ago and was fitted into the new activating mutation. It blocked both the initiating exon deletions and and EGFR active mutants, as well as a T790M mutation. What was also really good about this is it didn't hit the wild type receptor. So what happens when your biology is based on really sound scientific insight? Well, this is what happens. And this is the waterfall plot for every patient that was treated in those early development trials. If you see that horizontal black line, if you're below the line, your tumor is shrinking. And if you're above the line, your tumor is growing. Now, what I love about this is that I don't need a statistician to tell me whether my drug is working, which is always good. So from the very first dose, first patient, we saw the tumors melting away. It was very, very exciting, and this is the type of data we generated over the course of the past 2 years, where we've got now progression-free survival data for over 12 months. We have about a 90% control of disease, 64% response rate that has driven the very, very rapid moving of this molecule from development all the way to approval, and hopefully it'll get approved in Europe over the next 2 or 3 months as well. Now, what's important here, and why I'm mentioning this, apart from the fact this is a really nice case study of what good science can do and good development, is that we never stopped interrogating the science for this molecule. Even when we had 9291 in the lab, we started working on what we know was going to happen, which was new resistance mechanisms. And we wanted to start working on those before osimertinib was approved, so that we could start working on how to help the patients that basically were progressing on this drug in the same way they were progressing on Tarceva and Iressa. So we identified in our labs 3 different mechanisms of resistance to osimertinib. This first one was a pathway called c-Met. It's another kinase. It's a c-Met kinase. The ligand is HGF. And in a number of different tumor types, including this resistance people that have been on osimertinib, you either get a c-Met amplification or a c-Met mutation. It basically becomes the next oncogenic driver for these tumors. The good news is that we have a c-Met inhibitor in our pipeline. It's called AZD60914. And so we actually have taken patients that have developed this c-Met mutation, and we've now dosed them subsequently with AZD6094. And you can see one of those patients here. This is her lymph node. She's a lung cancer patient. She had an EGFR activating mutation. She stopped responding to the EGFR inhibitors. They sequenced a tumor, found the c-Met mutation, put her onto 6094. And straight away, her tumors melted away. So again, she would have had 3 to 6 months to live. She's been on drug now for 12 months and doing well. This is what continuing to study the science does. It enables you to think about how you can make these sequential therapies, and in this case, make the treatment much more long-lasting than would have otherwise been possible. The second resistance mutation, another pathway that you'll be familiar with, MEK. So MEK activation. A number of the tumors were generating activating mutations in MEK. And then the good news is, again, we have another molecule called AZD6244, selumetinib, that is also in late-stage development for KRAS-positive non-small cell lung cancer. So we now have a study that's going on with those 9291 patients that have developed MEK resistance pathways, where we put them onto selumetinib. And again, hopefully we'll show that we can slow or halt the progression of the disease for a period of time. We also found a novel mutation, a C797S mutation, and that's shown here. This is actually an experiment that this is actually in a patient that was dosed. And what we've done is we've collected tumor samples, serial biopsies, as the patients have been in the studies, and we've actually looked at how their tumor changes over time. What we found was, again, this other mutation that basically became an activating mutation that made the patients insensitive to osimertinib. And so one of the things that we did was actually then create a series of cell lines, and this again shows the power of genomics and the new gene editing technologies, because we use CRISPR to do this. We were able to do this in weeks as opposed to months and years. We generated cell lines that actually have each of the activating mutations that are important for driving resistance. So you can see in the top panel, we've got exon 19 deletion, which traditional EGFR inhibitors work in, as does osimertinib. The next one down has the T790M mutation. So now gefitinib becomes resistant. You can see osimertinib still works. With increasing concentrations, you can see that we're still seeing nice dose response. And then at the end, we've added all 3 mutations into a single cell line, and you can see now that both 9291 osimertinib is resistant, as is gefitinib. And so what that now gives us is a reagent, a cell-based assay, where our chemists can start to use this model to start to develop molecules that hit all of these activating mutations and develop the next molecule that will hopefully attack this mechanism of resistance. Now, the power of this is actually how CRISPR has enabled us to do this in weeks, as I said, and really, really quickly, so that we've started to work on this even while osimertinib is in development. So incredibly powerful. So that gives an example of lung cancer. Let me give you another example in DNA damage response, where again, it's an incredibly well-validated pathway, and defining the right patient population really gives you an in and an opportunity to really define a fantastic benefit risk for patients. Now, DNA damage response is incredibly important in tumors, and we've been working on this for many, many years. And what's good about when you work on an area deeply, it tends to always end up being more important than you think. So as tumors become more highly mutated, they become more dependent on certain DNA damage response pathways. So for example, if you have a BRCA1, BRCA2 mutation, the mutation that Angelina Jolie had, you become much more sensitive to single-strand DNA repair mechanisms, which are modulated by an enzyme called PARP. So PARP inhibitors work well for patients that have BRCA1, BRCA2 mutations, either in ovarian or breast cancer. But since that time, we've actually found that PARP inhibitors work in a much broader subset of patients with DDR mutations. And we've seen, for example, that they work also in prostate cancer, in non-BRCA mutated prostate cancer, but where there is other deficiencies in genes such as ATM and ATR. And what you can see here is a panel of molecules that we have that go after very specific DNA damage response repair pathways, whether they're single-strand breaks or double-strand breaks, that enable us to go after subsets of patients. So let me just show you some data from olaparib, and this highlights the importance of selecting the right patient population, not going after all of ovarian cancer, but going after BRCA-mutated ovarian cancer. And you can see here in the placebo arm, you can see this is a plot where the quicker you drop, the more progression of disease you have, or you can also see survival curves like this as well. But you can see the clear differentiation between olaparib-treated patients that are BRCA positive and placebo-treated patients. If you actually did this in all ovarian cancer patients, you'd see the signal diminish. It would get smaller. So actually your risk-benefit changes. So this is a drug that was approved last year in the US and the EU and is associated with a diagnostic test in the same way that actually osimertinib is. And is working much, much better than we ever expected because we're targeting the very important subset of patients that we think will respond the best to it. Now, the second molecule I talked about is a molecule called WEE1. This is a molecule that drives cells through catastrophic mitotic cell death, basically pushes cells through the cell cycle by inhibiting CDK1 and CDK2 and doesn't give the cancer cells time to correct any mutations in their genome. And what that does is it basically causes them to undergo apoptosis. Now, we ran a study initially in platinum-sensitive ovarian cancer patients, so ovarian cancer patients that respond to chemotherapy, and it worked, but it didn't work any better than chemotherapy alone. The really exciting data came when we went into platinum-resistance patients. So patients that when they're diagnosed maybe have a 10% response rate to chemotherapy, and it's very short-lived, maybe 3 months before their tumor progresses. And this is a study that was conducted by Jan Schellens at the Netherlands Cancer Institute. And what we showed was that we were getting a response rate in this resistant patient population of 40%. And some of those patients on the right-hand side, again, if you're looking at their tumors shrinking, their tumors are not shrinking by 20 or 30%. They're shrinking by 95% or 100%, basically becoming undetectable. And they've been on drug for more than 2 years. This is a patient population that would normally have 3 to 6 months to live. Absolutely phenomenal data, very, very exciting. And what we've been working on, again, doing serial biopsies, is trying to define the genetic code of those tumors, of those biopsies, to understand what's the population that is going to be most sensitive to our WEE1 inhibitor. There's lots of other things that we're doing this, because ultimately we've also got data. I don't know if it's in here. Let me just see on the next slide. Here we go. We're showing that we can combine it also with olaparib. This is a preclinical model. And this is a molecule where we've tested olaparib on its own in this preclinical model. It's a xenograft model where you basically implant a tumor into a mouse with a known genotype, and you look at sensitivity to your particular agent of interest. So olaparib or WEE1 both work in this model, but they work okay. When you compare the red line, which is the one right on the very bottom, you see what the combination of WEE1 and olaparib do together. It basically flatlines the tumor. And we stopped dosing in this experiment after 28 days. And invariably, when you do these types of studies, the tumors grow back in the animal. The tumors don't come back here. They basically we've cured the animal. If we can obviously translate this into humans, it becomes very, very exciting. So our WEE1 molecule will go into olaparib-resistant patients. It will go into subsets of patients that we've genetically defined that we think will be most responsive to WEE1. And it will also go into studies where we combine it with WEE1, with olaparib, sorry. And then finally, again, I'm not going to talk about immunology and immuno-oncology, but all of these approaches we think are making the tumors more immunogenic, and so we think will be very well combined with immuno-oncology agents that are also transforming cancer care today and are enabling patients to mount an immune response to their tumor that is giving us response rates and durability of response that's actually unprecedented in terms of what we've ever seen in melanoma and lung cancer, as well as a number of other tumor types. Okay, so that's 2 oncology examples showing you the power of good scientific understanding, patient stratification, how we've used a number of different technologies to really help validate and prosecute our targets as effectively as we can. Let me go into an example for COPD. So salt-inducible kinase. This is actually an early-stage program where we have some interesting biology, some interesting insights, and it's really trying to understand the role of macrophages in driving COPD. And we know that macrophages are important in COPD, in particular in the tone they have in terms of being pro-inflammatory versus anti-inflammatory. And there's some pan-Syk kinase inhibitors that have been shown that they can basically switch off the inflammatory state of a macrophage. And I'm showing you one of those molecules here, MRT 67307. And what you can see is when you dose these macrophages, these human-derived macrophages with this molecule, you can increase the levels of IL-10, which is kind of an anti-inflammatory cytokine, and you can reduce the levels of TNF, which is pro-inflammatory. So the question our scientists were interested in asking is which Syk kinase is the one that's important in driving this phenotype, and can we define it, and then as a consequence start a drug discovery program that goes after the very specific Syk kinase to give us the best risk-benefit in terms of tolerability as well as efficacy. So what did we do? Well, we took iPS cells, and we took them through a differentiation protocol, select them for CD14 to give us monocytes, and then for CD68 to give us human-derived macrophages, and we started to play with these in our in vitro assays. And actually, again, the bit that I forgot to mention is that we also engineered them using CRISPR again to express either Syk1, Syk2, or Syk3. So the experiment here is we're taking iPS cells that have been engineered, differentiating them into macrophages, and then working out which Syk is important for driving the anti-inflammatory phenotype. So the first bit of validation that we did was just to show that in these iPS-derived macrophages, we could see that same response I showed you in the first in vitro experiment. We can see a reduction in TNF production and an increase in IL-10 production. So these iPS-derived macrophages work like they should. in terms of the in vitro assays, and then using each of the six engineered cells, and I've labeled it here ABC, and I'm not unfortunately able to tell you which SIC it is, but we did find that one of the six, very specifically, was able to reduce or basically was responsible for showing that anti-inflammatory phenotype. So variant B, as you can see, is very different to variant A and C. And so that now has become a drug discovery program that is focused on the specific isoform of SIC that will then hopefully move into lead optimization and ultimately candidate selection and give us a much better validated molecule than having a pan-SIC inhibitor that will have a number of side effects and tolerability issues. The last case study is around BACE, an enzyme that I started working on when I was at GSK in the 1990s for Alzheimer's disease. It's a very interesting target, and again, I think it's one of the best validated pathways in Alzheimer's disease. The Alzheimer's world, as many of you know, is kind of segregated into two camps, the Baptists and the Taoists, those people that believe in beta amyloid and those people that believe in tau. I think the fields have converged actually over the past decade, and people believe that the pathways are actually quite closely intertwined. But many familial Alzheimer's disease mutations for early onset Alzheimer's disease have either mutations in the presenilin genes or the APP genes and cause a change in the processing of a gene called APP and the production of a toxic protein called beta amyloid. So the hypothesis here is that if you reduce the production of beta amyloid derived from the amyloid precursor protein, you reduce the levels of toxic protein in the brain, and you slow or halt the progression of Alzheimer's disease. That's the scientific hypothesis that you're trying to test. Now, there's also been some additional genetic studies that have come from Decode and Amgen looking at Icelandic patient populations, where they've also found specific mutations in the APP gene that confer protection from Alzheimer's disease by modulating the activity of BACE to be able to cleave the APP protein. So these mutations actually reduce the ability of BACE to cleave the APP and actually, again, make less of this toxic amyloid beta protein. So BACE is a target, whether you believe in amyloid or not, is a good target to do a scientific experiment, ultimately in the clinic, to validate or invalidate the amyloid hypothesis for Alzheimer's disease. So we've done a number of experiments preclinically where we showed that we have a very nice BACE inhibitor. This is actually is the structure of BACE. And again, this was actually, I remember working with David Simmons at GSK when we cloned this. And people have been working on this target since the 1990s, early 2000s. It's been a very, very difficult target to work on from a pharmaceutical perspective because it has a very large, shallow catalytic pocket, which means that it's very difficult to get molecules that are selective, potent, orally bioavailable, and cross the blood-brain barrier. So from a pharmaceutical chemistry challenge, this has been a huge, huge problem, which is why it's almost 2 decades later that the first molecules are entering phase 2 and phase 3. Many molecules have gone before and have failed. Now, one of the reasons why they've failed is, again, because we've been learning about the SAR in the pocket. This is actually one of the early shearing molecules that we made. It was actually a pretty potent BACE inhibitor. The problem was that it also inhibited a channel called hERG, which is found in the heart, which leads to QT prolongation. So again, this is a really good example of listening to your safety signals, because these molecules all went forward into the clinic, and they actually died because they had no safety margins because of QT prolongation. So we knew this was a problem. So when we were running our program, QT was one of our screens to make sure we didn't move a molecule forwards that had a hERG liability. So we kind of got rid of that before it became a problem. And so our chemists were very focused on that as one of the challenges for the molecule, for the program. So this particular molecule, just to give you an example of one of our molecules, again, it had a hERG assay in there, and it had over 1,000-fold selectivity versus hERG in terms of potency and ultimately exposure in humans. What that enabled us to do is to do the preclinical experiments and ultimately go into the clinic and demonstrate, and this now gets at the, are you hitting the target, demonstrate that in patients and healthy volunteers, we would reduce beta amyloid in the CSF of humans, not just in animals. So what you can see here is 2 different doses of AZD3293, which has been dosed over time, and you can see that we're seeing a nice dose response in terms of A-beta lowering of either 50% or 75%. So this doesn't mean that it's going to work. But what this does mean is I know that when I do my phase 2, phase 3 experiment, I'm going to have tested the scientific hypothesis. So if this fails, I know it's failed because the amyloid hypothesis is wrong, or the A-beta hypothesis is wrong, not because I wasn't lowering A-beta enough. And that's a good fail. I'm actually okay with that. What I want to be sure about is that I've tested the hypothesis. And so that's very, very important. The other piece that we've done with this, because if you remember I told you that neuroscience was not one of our core areas, is we've partnered. We've decided not to do this on our own. We've partnered with Eli Lilly, who are a company that do have this as one of their core areas. And what this does is it buys us the best of both worlds. We have a molecule that we're very excited about, but we're working with a partner whose core bread and butter business is neuroscience and neurology and Alzheimer's disease. They have a huge investment here. And the fact that they wanted to partner with us on this molecule, one, is a testament to the quality of the molecule, which is great. But two, we share the risk in terms of the R&D costs, which are huge, many hundreds of millions of dollars in phase 2 and phase 3. And ultimately, we share the reward as well. So as far as I'm concerned, and I think our company is concerned, this is a win-win and a great way for us to move a neuroscience asset and enable us to work in a non-core area when we're trying to focus most of our resources in those 3 core disease areas. Okay, so that gives you some case studies and the types of things that we do to understand target validation. Let me talk a little bit about technologies and how they're enabling and changing our environment in terms of research and development. And I've highlighted some of the technologies that I think are making the biggest difference to us as an organization and I think to our industry. Intractable targets, and again, I remember my days at GSK and actually Wyeth and Pfizer, where the chemists would tell me, it's impossible, Mene. Don't work on that target, it's impossible. BASE actually was one of those targets. The program at GSK got closed in, I think 2000, in the mid-2000s. It was an impossible, intractable target. Don't waste your time. One of the goals I've set my organization is to work make sure that we're picking the right target and not worrying about whether it's tractable or intractable, and then finding solutions to how to get molecules that modify that pathway or that biology by either finding things that are sort of peripheral to the target or using technologies and platforms that enable us to go after it. So for example, if I look at the collaborations with XChem using DNA-encoded libraries, which are sort of fragment-based libraries where we have billions of compounds, and you can do the screens in Eppendorf tubes, it enables us to do so much more than using a traditional high-throughput screen. When we look at what we do with Peptidream, where we're using cyclized peptides to, again, define molecules and pockets in the targets of interest that would actually traditionally not be amenable to small molecule drug discovery. We're really trying to open up the target space to enable us to work on whatever targets actually our scientists want, not the ones that are derived by the capabilities that we have from traditional small molecule chemistry. And Heptara is actually, which is a local company, again, we work with them in the GPCR space, and they've enabled us to identify leads to a number of GPCRs that have traditionally been intractable and have failed traditional high-throughput screens. So the technologies that we build constantly need to be evolved and adapted to enable your organization and your scientists to not be put off by difficult targets where the biology is very strongly validated. Another way of doing this and of enriching what we do, which again is would have been heresy, I think, 10, 15 years ago, is to exchange compound collections. Again, I remember in previous lives where I've actually tried to do something like this, where I was, the chemistry collection was the crown jewels of the company. To me, it's a starting point, a tool, something that enables our scientists to define better molecules, optimize molecules, and ultimately discover medicines. So to give you 2 examples, a couple of weeks ago, we announced a collaboration with Sanofi where we exchanged 210,000 of our molecules. So they took 210,000 molecules in their library and gave them to us. We took 210,000 in our library and gave them to them that were pre-specified based on our chemists using chemoinformatic techniques to identify chemical diversity that Sanofi had that we didn't, and vice versa. So all those 210,000 molecules that we've exchanged are molecules that enrich our library and enable us to hopefully find better molecules for the high throughput screens that we run. With Bayer, we have a different collaboration, where if we run a high throughput screen and we fail to find a molecule, they screen against their library for us, the same target, and again, vice versa. So we have about, I think, 5 screens a year that we can do, where we run our screens, we don't get anything, we give them our assay, they run it against their compound collection, and we see what they get. Because our libraries are very distinct, very different, again, we've been both able to yield hits and leads that we wouldn't have been able to do if we were working as a silo or in isolation. So these types of collaborations, I think, are quite novel, quite different, and really highlight how we're trying to work much more together as an industry to enable us to better prosecute targets that we're excited about. Platforms. Talked about small molecule and fragment-based discovery. We're also working very, very hard in the non-small molecule space and in the oligonucleotide space. So we have 3 key collaborations. One with a company called ISIS, where we work in antisense oligonucleotides to block the expression of any messenger RNA that we're interested in across cardiovascular disease, metabolic disease, and kidney disease. We have a collaboration with a company called Regulus, working in the microRNA space to, again, modulate the expression of a variety of genes or microRNAs, again, in the cardiovascular metabolic space. And finally, with a collaboration with a company called Moderna, where we're able to express modified RNAs to express any RNA in cells again, to ultimately make any protein, whether it's a real protein or an engineered one, in any cell type that we want. And that's both in the cardiovascular and oncology space. And I'll show you some data of our most recent molecule. This is a molecule that's going to be entering clinical development next year, and it's using the modified RNA technology of Moderna. And this is targeting a protein called VEGF-A. Now, VEGF-A is a growth factor that is very well understood to have a role in blood vessel formation, endothelial cell differentiation, as well as the production of cardiomyocytes and the differentiation of cardiomyocytes in the heart. The challenge with VEGF as a therapeutic is that when people apply recombinant VEGF, it causes edema, and it doesn't have a great half-life, and locating it in the right area is not easy. What we've done here is actually inject modified RNA into the heart of an animal that's had a myocardial infarction, and we've been able to show that it behaves much better than recombinant VEGF. If you just look at the middle panel, you can see the blood vessel formation in the top, which is saline treated, or luciferase modified RNA treated, versus the bottom panel, which is the VEGF-A. And you can see the increased complexity of the blood vessels in the area of the heart. So we're basically regenerating blood vessels. We've actually shown that we're regenerating arteries in the heart. And it makes it, and actually that results in improved cardiac output and cardiac health, either by pathology or functional readouts. And this was published in Nature Biotechnology a couple of years ago. So this molecule, we've repeated many of these experiments, we're doing it in large animals now as well, has the potential to treat heart failure. And using a therapeutic platform that, again, 2 or 3 years ago was unheard of, and I think has great potential. I've mentioned the capabilities that our MedImmune group has, and again, I think this is one of the strongest protein engineering groups that I've ever experienced in my career. And when I look at some of the things they're doing, this particular example is, again, taking a well-validated target, HER2, which we know is important in driving a number of different tumor types. There's validated therapeutic antibodies out there. But what we've tried to do here is develop a HER2 that is even more powerful than pertuzumab and TDM1, which are the currently available drugs to patients. And what this is actually it's a biparatopic, bispecific antibody. This basically binds to different domains on the HER2 receptor, and what it enables the antibody to do, and it's also got a toxin attached to it, so it actually kills the cancer cell that it latches onto. This bispecific nature of the antibody enables it to not just target the cells, and you can see in the middle panel, you can see trastuzumab, which is the standard approved drug. You can see our biparatopic, so you see it binds to cells very nicely, but look at the difference on the right-hand side after 6 hours. You can see how it's being internalized much more effectively than trastuzumab, and because it's got this lethal toxin engineered onto it as well, it means that we can kill those tumor cells very, very specifically. And then when you compare in a xenograft model, again, the activity compared to trastuzumab or TDM1, you can see that our animal knocks the tumor out of the park in terms of regression, and the tumors don't grow back. These animals have gone on way beyond that, and basically, again, these animals are cured of their of this particular tumor xenograft. So very exciting data. This molecule will hopefully be entering clinical development. And again, well-validated biology, where we're using protein engineering to devise and design something that's quite different to standard of care, and hopefully will have better activity. Now, human cell technology is, again, another area that has been really evolving over the past few years. The use of induced pluripotent stem cells and human-derived cells to create much better models that hopefully are more predictive, both in terms of target validation but also toxicity. So if I think about the cells now that our safety group is using, whether they're being complex cardiac human cultures, human hepatocytes, kidney cells, you name it, we've got human versions of it, and we're starting to use this in ways that is really changing how we do our preclinical models and actually giving us a much better indicator than the traditional rodent and dog models that we use for toxicology. Now, our long-term aim, and this is just showing you some examples of what we're trying to do, this is a little bit science fiction-y, but we have individual cultures actually out there for each of these tissues. So you can see each of the different organs. You can see on the left-hand side the beating cilia of our human lung culture. We're working with the Weiss Institute here to develop in vitro models of the lung. You can see on the right-hand side, we've got various neuronal cell types, and we're creating complex neuronal cultures from patients with schizophrenia, with autism, working with Danny Weiberger in Washington. You can see the gut cell. This is really cool. So that's a gut cell that's basically developing over a period of weeks, that's forming all the microvilli and all the complexity that you'd see in a normal gut, human gut. And we actually use this for one of our programs with a preclinical species, the rodent and the dog, were telling us we wouldn't have a window. And when we put it into the human assays, and we actually tested it against other molecules that have been in the clinic, it showed us actually that the preclinical species was more sensitive than the human would be. And it enabled us to create the margins to go forwards. So again, these are the types of things that we're doing at an individual level, but the ultimate aim is to connect these things together using microphysiological systems, so you can get, for example, lung connect to heart, connect to liver, and actually see how you can create a human body on a chip, not just an organ on a chip. It's very exciting, and again, we're working with some pretty cool scientists across the world to help us do this. Personalized healthcare. I've mentioned this a few times and given you some examples of how important it is. We're not a diagnostics company, but we've launched 7 diagnostics this year, more than any other company actually in the world. And over 80% of our pipeline, if you go back to that 5R slide I showed you, where one of the key R's is to find the right patient, 80% of our pipeline has a personalized healthcare approach for defining the right patient population. It's so important. For me, it's one of the key things that has transformed our productivity and our ability to not just move drugs into late-stage development, but also to get them approved. We were the first company to get a circulating tumor DNA diagnostic approved for EGFR-driven lung cancer. So this means that a patient with lung cancer can come in, have a blood test done, rather than having a biopsy done. Many lung cancer patients can't have a biopsy because their tumor is inaccessible. So they have a blood sample. They get sequenced to see if they have an EGFR-activated mutation. If they do, they go on to the appropriate drug. We'll also be the second company to launch a circulating tumor DNA diagnostic with osimertinib. So even though we're not a diagnostic company, we're really shaping the diagnostic landscape, working with a number of different partners. And over the next 5 years, we'll have over 30 launches that have got diagnostic, companion diagnostics associated with each and every drug that gets approved. That's a very, very important cultural shift, and I think is really changing how we have been working, how we are working in the organization. Finally, just to mention a little bit about open innovation, because it's something I've been very passionate about since I joined Astra Zeneca, and been in a position to influence more so than I ever have before. We often hide again behind the confidential nature of our business, which I think is actually, very flawed in terms of its thinking. I want us to be sharing as much of our data as we possibly can. Obviously, I need to protect IP or protect the head start if we have it for some of our science. But generally speaking, we need to be sharing data as much as we can with people that we collaborate and people that we partner with. So we've created this website. And when I look at the tools that we have available to anyone in the academic community, and we started this off with the collaborations we have with MRC and NIH when we made 22 of our clinical compounds available for clinical or preclinical investigation. We gave them the molecule, we gave them the dossier, and basically whatever great ideas they had, they were funded by the MRC or the NIH. And we have many molecules that are currently in development today through this open innovation pipeline that are going to be reading out over the next 2 to 5 years. So for example, we have an oncology drug that is currently in phase 2 development for Alzheimer's disease with Steve Stripmatter at Yale. We have a molecule that was in development for esophageal reflux that failed, that is now in development for chronic cough. I can hear a few of you coughing. So these types of things, I think, are very powerful ways of generating value, not just for us, but for investigators to start new biotechs up and ultimately create more value than would be inherently created if we just kept the assets to ourselves. We have the same type of tools in the preclinical space, where we make many of our compounds accessible for preclinical investigation. We make our assays available in terms of DMPK assays, toxicology assays. So if an academic has a molecule, they want to find out if it's toxic or if it's got certain physicochemical or PK properties, we'll do the experiment for you without necessarily needing anything in return. We want to try and help the community move their programs forwards. We have R&D challenges. When you look at the types of things that have started up as a consequence of this, 80 preclinical experiments, 20 clinical trials, this is generating nearly $45 million worth of funding. This is generating basically a parallel universe of R&D for us that could yield any number of exciting opportunities, whether it's for us or for a partner. And it's something that I'm very proud of and very excited about. And if you want to find out, the website is there on the bottom left. And then finally, I couldn't avoid but mention the most recent picture of our new building. One of the reasons we've moved down to Cambridge is because we want to be surrounded by fantastic science. Here at the EBI and the Sanger Centre, at Addenbrooke's, LMB, Cancer Institute, the science in this part of the UK is absolutely phenomenal. And it gives us access, obviously, to science in Oxford, in London as well as around the UK. But this is going to be a phenomenal place. And the whole culture, the whole ethos of the building that we're building is one about being transparent, open, collaborative. You can walk through it. You can use a coffee shop. It's not going to be surrounded by big barriers and look like a traditional pharma building. The idea is that it feels open in nature and style and has a collaborative approach in terms of how we interact with the surrounding community. Hopefully still be ready by the middle of 2017. We had our 1,000th employee recruited into Cambridge last week, so we're well on track. And actually, the great thing about being here early is that it really gives all of our science a chance to get very well networked with the Cambridge academic community as well as the community in the surrounding region, which is really good for us as a company and hopefully good for the scientific community as well. So what I hope I've done over the past 45 minutes or so is give you a flavor for why we think being collaborative is critically important. Talk about the fact that we've created an environment where our scientists have to publish, where they have to thrive, where we challenge them, and where they also challenge conventional thinking and take the impossible and try and make it possible. And with that, I hope that we can really demonstrate what science can do. So I'll leave it here. Thank you all very much for your attention.